simply having it, the regulations state that SRY is used as a proxy to determine if you have a Y chromosome or not. And that's their bottom line. If you have SRY, then you're going to have a Y chromosome and vice versa. But that's the part that's just not 100% true, because you can have or not have SRY, and that's irregardless of having a Y chromosome or not.
Hi, Shanie. It's my pleasure to have you on Evidence Strong Show for the second time. And please briefly introduce yourself.
Yes, Alex, thanks for having me. It's nice to talk to you again. So last time we talked about sex differences in exercise physiology, and so I introduced myself then, but I'll say again that I'm a researcher in genetics and molecular biology and how it relates to sex differences, both in health and disease. And also in the past, I had a stronger focus in exercise physiology, and I'm an 800-meter runner myself, so I guess even more related to this topic. And yeah, I'm in Melbourne at the Hudson Institute doing a postdoc and finished my PhD in epigenetics about 4 years ago.
Amazing. So today we talk about DSD athletes and transgender athletes and the policies and how they relate to research. So let's start with some kind of definitions. What DSD athlete is and transgender athlete and how they differ.
Before talking about DSD and transgender, I'll define sex and I'll define gender. And I think that those are commonly used interchangeably in society, but in biology, there are two separate words with separate definitions. So sex, we call the biological sex, and the two main categories are male and female. And I'm keeping it simple for the sake of the definition, and we'll go into more details after. And this is determined by our sex chromosomes. So a female typically will be XX, and having XX chromosomes means you have a certain set of genes, and those genes lead to development of ovaries. On the other hand, you have XY individuals, that means they have an X and a Y chromosome, and having the genes on those set of chromosomes lead to the development of testes in typical males. And so I guess to simplify it, the way we define biological sex in biology is based on the phenotype or the gonads. So if you have testes or ovaries, so basically your reproductive organs. And that's how we kind of refer to it, at least in the field that I'm in and the way that we're approaching that biology. Gender, on the other hand, is a social construct. So it's the gender is what one identifies with or perceives themselves as. And that more broadly, the two categories is man or woman. So to kind of use the terminology correctly, which it's often not, and even in a lot of peer-reviewed papers and a lot of really high-impact papers, people will still interchange them. But if you're talking about what is actually happening from a biology perspective, sex is male and female, and gender is man and woman. And that means that comes up a lot in research is that mice don't have a gender, they have a sex, because we don't know, mice can't tell us what they identify as, right? So they can be male or female, they can't be, they don't have a gender. And that's, again, to keep it simple. I won't go into all the different variations of gender identity. So I gave you just the two, man and woman, but yeah, I won't go into the details of all the in between, because we study biology, so we're interested in what's in between male and female, but we're here to talk to you about biology, not about the social construct, so I'll stick to that. And then how that relates to DSD and transgender. So DSD stands for differences in sex development. It used to stand for disorders in sex development, but that was changed. It used to be termed as intersex, but that's also less commonly the medical term. So now we go with DSD, which is a difference in sex development. So a lot of what my lab at the Hudson does is study those exact pathways of sex development. So how does an XX embryo lead to the eventual sexual characteristics? And what that means is about 1% of the population, which is a lot more than what people tend to think, and that is kind of commonly thought, is born with a DSD. So that means they present with sexual characteristics that are not 100% typical to male or 100% typical to female, but are somewhere in between. And that's a really big range and a really big spectrum. So you can have anything from a partial sex reversal to a full sex reversal. So what we mean by partial is you are XX, you're presenting as a female, but you have some characteristics that are male-like. And that's why we use the word typical, because 99% of the population, that's how a male develops. 99% of the population, that's how a female develops. So this is the typical pathway if everything is coordinated in a specific way. And variations to that pathway will lead to a difference in sex development. And that can be at several stages along the pathway, therefore leading to this spectrum. So that was partial sex reversal, meaning you still have the sex chromosomes that your phenotype or your gonads are presenting as, but maybe you have some variation. And then you have full sex reversal, where you can have an XX chromosome makeup, but you're actually presenting entirely as a male and having male characteristics. So there's a range and everything in between. And so what my lab does is try to identify the genes that are, or the variants in the genes that are causing those differences in sex development. So the main gene that was discovered several decades ago was SRY, the sex determining region on the Y chromosome, is what SRY stands for. And it's a gene that lies on the Y chromosome. And generally, typically, if it's present, it leads to the development of testes. So it's kind of like, as a simplistic statement, if you have SRY, you'll develop testes. If you don't have SRY, you won't. But that's a very simple statement because the pathway is very complex. There's a lot of parts of the pathway that are already known. So which targets there are upstream or downstream of SRY. And there are also inhibitory pathways to then inhibit the development of ovaries at the same time. So it's a coordinated pathway. It's almost like you start with what's called the bipotential gonad, and it kind of has an unknown fate at that point. And it depends on the presence of a multitude of different genes in the pathway. And if the right genes are present at the right time, so they're expressed at the right time, that bipotential gonad will differentiate into a testes. And if the right genes are present or not present at the right time, that bipotential gonad will differentiate into the ovaries. So it kind of has these two pathways that it can go down. That said, because it includes a number of genes that need to be coordinated and turned on at the right time in the right place, right in the gonad, then variations, whether it's at this point in the pathway or at this point, then all of a sudden you could find yourself coming towards the other pathway. So that can happen at multiple different stages, and that's where the complexity comes in. So there's about 50% of cases, and I'm giving a rough estimate because it depends on which type of DSD, if we're talking about an individual that's XY or an individual that's XX, but at least 50% in both categories have variations in genes that we haven't yet discovered, or variants that we haven't yet discovered. So about 50% of the cases that present will, if a baby is born and presents with ambiguous genitalia or ambiguous sex characteristics, so kind of a mixture of both, then they usually undergo a DSD gene panel, which has 138, so approximately 140 genes that have been confirmed and identified to lead to DSDs. If they test positive for any of those specific variations of those specific genes, then they have a diagnosis and they can be advised with their treatment options, or kind of the way that DSD tends to develop in people. And then the other kind of 50+% don't fall on any of the genes on the gene panel. And that's the patients that we often get. So we'll get patients that are called panel negative. So none of the genes that cause DSDs that we know of don't explain their DSD. And then we perform a mixture of whole exome sequencing or whole genome sequencing with a combination of other methods to identify the potentially causing variant or that DSD.
Is it possible that it's not obvious at birth and the person gets to know later? And if yes, what circumstances would bring this topic about?
Yeah, so because it's such a spectrum, there's a lot of answers to that question, because depending on the type of DSD. But as an example, so the three kind of main types of DSDs that we categorize into are, one is sex chromosome aneuploidies, which means you basically have a different number of sex chromosomes than what is typically found. So instead of having XY or XX, you have either XO, which is Turner syndrome, so females that have just one X chromosome, and you have XXY, which is Klinefelter syndrome, so males that have two X chromosomes and a Y, and you have Jacob syndrome, which is XYY, which is more rare, and that also leads to male development, but with two Y chromosomes. So a good example of individuals finding out later on is with these sex chromosome aneuploidies, because they have some mild phenotypes, and again, it ranges depending on the individual, and what we call them mosaicism, so some cells can have just one X, some cells potentially have a combination. So depending on that individual's present genetic condition or kind of genetic makeup, they will present with different symptoms and potentially at different stages of life. So often what will happen either with Turner syndrome or Klinefelter syndrome is these people have problems with conceiving or problems at fertility. And upon fertility investigation, we'll find out that they have one X chromosome in a female or potentially two X chromosomes in Klinefelter men. So that's one example of how that can be found later on. And then back to the other two types of DSD. So we talked about sex chromosome aneuploidy types. Then there's what we call 46XY and then 46XX. And what that means is that the individual has the two sets of sex chromosomes that are typical, right? Either XX or XY, but they have a partial or a full sex reversal. So a variation to their development, meaning someone that's XX isn't presenting phenotypically completely as an individual who is XX, and vice versa with XY,
Wow, super complicated.
to say the least.
That's good. I really wanted to show it. Why do we care in sports whether the person developed typically or atypically?
In the context of sports, and this is where, yeah, it's complicated in biology, it's complicated in what we study, and so that obviously translates also to the sporting regulations and to the definitions in sport. So I'll go through kind of just a rough history of what's kind of happened. So update on the DSD participation, and I guess I'll keep my wording a bit vague because I'm not here to point fingers at anybody or at any body or organization. I'm a biologist or interested in the biology, and I'm here to discuss the facts of what we know and what we don't know, and not to give my opinion on what I think the regulations should and shouldn't be. So the saga with DSD participation is not new. It's something that's come up in history, but it really blew up around 2009 after the 800 meters in the World Championships. And since then, it went through a number of different regulations. So first, it was decided that females that wanted to compete in events from 400 meters to the mile in athletics had to have a testosterone concentration below 10 nanomoles per liter. That concentration, I'll talk a little bit about the testosterone aspect. That concentration recommendation or restriction then moved down to 5 nanomoles per liter, and then it moved down to 2.5 in 2019. So, and that's where it sits right now. And there's a lot of debate around that, but what that meant is that females that had a DSD that caused them to have high testosterone levels were required, if they wanted to participate, to take medication to lower their testosterone levels below that allowed threshold.
Could you just, my audience may not be quite familiar what the normal, maybe typical ranges should be for females and for males. Why 10, 5, 2.5? How it relates to male levels?
So males have several folds higher levels of testosterone, a normal, a typical male and a typical female. And there are conditions in females, and that's what I was going to get to and on the complexities in it, because there are conditions in females such as PCOS, so polycystic ovary syndrome, which can lead to those higher levels, but it's a trait that we call sexually dimorphic, which means it totally doesn't overlap. So there are traits that have sex differences, for instance, height. If you look at a graph, it's kind of like this. Like, yes, on average, males are taller and females are shorter, but there's going to be an overlap. It's not like there's not males that have the same height as a female and vice versa. But when it comes to testosterone levels, it's like this. Like you can take someone's testosterone and you want to know if they're if they're a male or a female. We're talking about. I keep using the word typical to keep that in mind. So yeah, it's many folds higher in males.
Okay, so if that's the case, why 10, 5, and 2.5?
Yeah, and that's what a lot of the debate and criticism has been around, because those numbers, at least in science, whenever there are thresholds, we always see that as a little bit arbitrary. We choose a p-value of 0.05, but some people choose a p-value of, and a p-value is just how significant something is in science, right? Of 0.05 or 0.005 or 0.1 or 0.001. So, choosing a threshold that's going to decide, if you're above or below that, always is going to have a sense of subjectivity to it and, I guess, arbitrariness to it. And I think that's why the numbers were adjusted every few years, is because there really wasn't evidence to support that threshold. So there were studies done trying to measure performance in females and their levels of testosterone and correlate levels of testosterone to performance. But we know that performance is so multifaceted. So how can you, single that one thing and say, oh, this person has high testosterone, therefore they have better performance, when there was maybe another person that had that same level and they had lower performance. So it's not a black and white. The levels that were decided were debated on, and that's why they kept coming down. And the data that was out there to support that threshold was very weak. Yeah, Yeah, and in addition to that, there's different reasons for having high testosterone, and there's different ranges. So if you have PCOS, and it's also all about the receptors as well. You can have high testosterone, but is it bioavailable? Is your body actually able to have the sensitivity in the receptors to respond to those higher levels? So it's a combination of a lot of different things that are going to present differently in an individual depending on the reason that they have that high testosterone and the background of everything else that they have. So there's kind of not been any single study that's been able to, really answer that question fully, because most studies and most of what we know about testosterone historically was done in studies in males. And now there are more studies coming out in females, and we'll get to transgender gender affirming hormone therapy, but looking at the effect of when you take exogenous, so outside of your body, testosterone and what that does. But when it comes to having high endogenous testosterone levels that have high variability and a multitude of different functions, it's really hard to answer that question of exactly at what threshold do you get a performance benefit with testosterone in females. It's just not really well known, and the data is not there to support that.
Okay, so if we think DSDs, we understand now that it's a spectrum and the things may diverge from the typical pathway on many, many levels. So now in terms of sport, the different DSDs will have different presentations in terms of potential performance improvement.
Exactly. And because it's 1% of the population, and in that 1%, that splits to, a certain percent being males and a certain percent presenting as females. So that's going to be even less. And then you have the number of those people that actually ended up in sports or having, skills in a specific sport. So that reduces it even more. And then you have the ones that they have variable presentation. So what are the genetic causes? And then it splits it up even more. So you have a really small number of people to represent, each one of those different categories, or however many there even are. But the point is to say that when it comes, so I didn't mention, so the latest ruling is what I was going to add. The latest ruling that was just now before the Tokyo World Championship, so just in September, was that the testosterone for competing in the female category needed to be below 2.5 nanomoles per liter, so that stayed. And individuals who wished to compete as a female needed to undergo a test to see if they have the SRY gene or not. So we talked before that the SRY gene is one of the key members in the pathway to determine if you develop testes or if you develop ovaries. But, and this is where the big but comes in, and where the DSD researchers are kind of trying to, I guess, make it clear that it's a lot more complex than that. And it's not just one gene that decides if you have testes development or ovaries development. So there are what we call SRY negative cases and SRY positive cases. And that means that, for example, an individual that is XY could not have the SRY gene and could have crazy things happen in genetics. Genes can get moved on to different chromosomes, and chromosomes can rearrange and split up in meiosis and separate strangely. Like, a lot of strange things happen. And simply having SRY or not having SRY doesn't determine the sex development 100%. It explains a lot of the cases, but not all of the cases. So that means that you can have a female, so an XX chromosome complement, but there can be a mutation in another gene, such as SOX9, and that can lead to some male sex determination pathways. So that's just an example of why this SRY simple gene test to decide if a female can compete or not doesn't actually cover the range or the spectrum of conditions, because there can be mutations. Well, there are mutations in other genes, ones that are known, but also ones that are not known, that will lead to male sex determination in a female, and vice versa. So a simple test is just, well, the biology is complicated. And, where do you draw the line for female enough? We don't even know that in biology. So I don't know how you would draw that line for sport. It's just too complicated. So I guess the other detail to add to the ruling that was made back in September is that women that were XY but are presenting as females that have what's called androgen insensitivity syndrome are allowed to compete in the female category as long as their testosterone is at a low level. And this is because they don't have the receptors to actually respond to that. So that was things that are based off data and rulings that are based off of science. So it's nice to see when the policies and guidelines do have some science rationale to them because that was argued. Well, they have higher testosterone, but they can't actually use it, so it's not actually helping them. But the thing that still stands that is not completely aligning with the science of DSDs is the fact that now the SRY gene is deemed the, be all know all to if you have a male or female sex development. And we simply know that that's not that simple and there's other genes involved.
Okay, should we move to transgender athletes?
Yes. So I have much less to say about this because we do study a little bit in my current lab and my past lab, but it's not currently something that, how do I say it?
No, but I think we need just to complement the topic of the athletes and to make it maybe not full picture, but maybe a little bit more rich picture. So we have these athletes where the genes and the development are varying from the typical, and now we have transgender athletes who at some point decided that they present or want to present as the other gender. So that has consequences in terms of fairness and participation in sports. So from that perspective, who are transgender athletes and how they differ from DSD athletes?
Yes, well said. So I think what's important to understand with transgender athletes and the decisions that they make around their process for if they want to perform gender affirming hormone therapy or not is understanding what administering hormones does to the body and to performance. So two kind of main things in that to understand is what we call organizational and activational hormone effects. And what that means is organizational is the lifelong exposure to a specific hormone level. So having high testosterone in males or higher estrogen in females means that there are things that will happen in the body, in the structure in your physiology that are irreversible. So a good example, a well-known studied example, is that there's a very specific time window in embryonic development that having high testosterone at that time leads to development of a specific region of the brain to be bigger. And that region of the brain is, again, in typical males and females, is always larger in males and smaller in females because there's that critical window of testosterone exposure. And if you treat developing female embryos with testosterone during that critical period, they will develop the male type size for that brain region. But if you give the testosterone later on outside of that critical period, then they won't. That brain region will develop as it does in females. So it's to show that there are critical periods in development that rely on specific hormone levels, and either having or not having them at the right time is going to confer sex differences that remain for life and aren't reversible. So growing up with a certain hormone level and having that exposure throughout your in utero and throughout your life is going to set up some differences that are not going to be reversed. That's what we call organizational. The activational hormone differences means that those are kind of the acute effects of hormones, and those can be reversed. So you can have, taking testosterone can have some immediate effects on your cell signaling, on your cell pathways that respond to that. And if you then remove that, then that pathway then stops that signaling or, goes back to what it was before. So there are effects that are long-term and irreversible and that happen from long-term exposure and effects that are reversible. So I think understanding that, and the field is still in its infancy in understanding exactly what the long-term effects are of gender affirming hormone therapy in transgender individuals. So I think it's something that's quite a hot topic right now. And I think that we're going to see a lot more studies coming out on it in the coming years. So that's something to keep your eyes open for. But a lot is looking at, what risks or what immune differences or what are the effects on the physiology and non-performance that are going to come about. So it's trickling in, and we are seeing a little bit of, the effects of strength performance if you take testosterone, if you take estrogen, and you do see an increase in strength in specific tests with testosterone and a decrease with reducing testosterone and increasing estrogen. But I think that there's just a lot still left to understand there. And yeah, there's also important to take into consideration how it affects the rest of your health. So there is an increase in risk with having thrombotic events, as well as a lot of a few other cardiovascular events, and having specific, genetic predispositions to those events might exacerbate that. So it's very much still in the process and young in understanding the exact effects of taking exogenous hormones for a long period and what that does. So I think when it comes to transgender athletes and the decisions that they can make for themselves, is that just having the knowledge of what we already know and what the effects can be for that person will allow them to make the decision of, what they want to do, because there are the regulations set out, which is having the testosterone levels at a certain level. And if they want to compete in the female category, then that is something to consider, because that's something that they have control over, as opposed to the DSD athletes, which is different because that's made endogenously. It's made from within. And it would be taking something external to reduce those levels, while in transgender athletes, they can choose, right, to change those levels to be higher as opposed to lower. So yeah, I think when it comes to transgender athlete participation, it's less complex and there's more power in the individual's decisions that they can make in order to confer to the guidelines and then be able to compete in the female category.
Okay, so what I'm hearing is from the embryo, there are differences between female embryos and male embryos and how they develop. And these differences grow with the long-term exposure to the typical hormonal profile the female or male will have. So transgender athletes are different fundamentally, and there are differences that cannot be changed or controlled for using the gender affirming hormonal therapy. So that's obvious on the base of the science. Now, how much it influences performance, that's where the gray area is and where the investigations are. Am I understanding correctly?
Yes, I'd say so. I would say gray, it's not as gray as DSDs, let's put it that way. So we know what taking exogenous hormones can do to your performance immediately, but I think we don't fully know so exactly what those hormones influence. But I think it's, even intuitively and understanding and looking at a male and a female athlete and the size difference and all the other sex differences that we talked about in the previous recording, is there are going to be established sex differences that are not going to be able to be reversed. So yes, taking exogenous hormones or gender affirming hormone therapy will shift those numbers a bit, strength wise, performance wise, but the difference between male and female performance is so large that, that's kind of how I see it. So there's more left to understand. I'll agree with that, but we know enough to know that the lifelong exposure to hormones, and even not lifelong, even if people undergo, hormone replacement therapy earlier on, there's still in utero differences and going through puberty that are going to be, irreversible in a sense.
Do we know anything about for the transgender individuals, maybe not necessarily athletes, because I don't think we have enough research to draw on that, but how the gender affirming hormonal therapy influences risks of developing health conditions or diseases that are typically correlated or are more common in one or the other sex in typical lifelong exposure?
I love that question. That's a great question. I think it's fascinating to when we are able to hopefully one day answer that. That is very much kind of connects to the a lot of the research that we're doing, which is understanding the basis or the reason for sex differences in conditions. And we talked about last time, I think, that there are diseases that are more prevalent in males, or the symptoms are more severe in males, and vice versa in females. And a lot is still understanding what are the drivers of that, if it is more sex chromosome genes or if it is more hormones. And the more that we've been able to answer some of those questions around some of those diseases. And I think that will tie in really interestingly with your exact question, which is which diseases are we potentially protecting or becoming or exacerbating the risk to in males or females who are undergoing hormone replacement therapy. I don't from what I know, there's not enough out there to answer that question, but I think that's going to come out as a really interesting question to answer, but for transgender individuals, but also for for the rest of the population as well. Yeah, I think as it does relate to the nice intersection between, that we're talking about performance and sports, but also about health, because of course, at the end of the day, we want to be healthy so that we can compete in our sport, right? So knowing the broader implications of hormones at the different stages and what it means to be taking them at different stages for your health is, yeah, a growing body of evidence, I would say. So there's a bit known as what I just mentioned, but there's still a lot left to understand with that. I don't think there's going to be one fair solution that is inclusive of all and fair to all because there's just so much complexity in it. So I think the more we can study these conditions and how they relate to performance, the more we can design experiments that really help us to understand what testosterone is doing in females and in female muscle and in female performance. The best that we can design those experiments and answer that question and have a threshold that maybe is really based off of strong scientific evidence. You would say you would have a gene panel and you could look at all the DSD genes and then you could make decisions. But yeah, there will be individuals that fall in that in between spectrum. And that's a lot of the issues that also individuals with DSDs will experience with making medical decisions with their conditions is, if you're somewhere in the middle, how do you choose it's hard to draw a line in biology in that sense. So I think it's hard to draw a line in sport as well, which I think I kind of repeated as before. But I think, yeah, my simple answer is that it's too complex, and there's in my opinion, there's just not going to be one solution that fits for all. And the more that we can base it on scientific evidence and strong scientific evidence that's robust and not biased, it'll be the most fair that we can have for the most people. But yeah, I think that's the best that we can do.
All right. Is there anything I didn't ask about but you think it's important to bring while we on this topic today?
Yeah, as long as you think that I explained the sex development pathways and how it doesn't solely rely on SRY, because that was the main takeaway that I wanted to get across here, because I know from friends who competed in the last world championships that, they did their SRY test and all was good and well, but the athletes don't actually understand the meaning of it, I think, at a deeper level. And not that I expect people to, because it is complex and, the average person isn't studying sex development pathways in their free time. But I think that when you're subjected to take a test that's going to tell you something about yourself, and when all of your peers that are competing are subjected to take that test, I think that there should be more information out there to understanding the complexity of it and what it really means to have an SRY test. Because I don't know if I touched upon this, but simply having it, the regulations state that SRY is used as a proxy to determine if you have a Y chromosome or not. And that's their bottom line. If you have SRY, then you're going to have a Y chromosome and vice versa. But that's the part that's just not 100% true, because you can have or not have SRY, and that's irregardless of having a Y chromosome or not. So I think, yeah, as long as my main point came across, which is that, the test that's currently the regulation is trying to make something gray, black, or white, is what I'd say at the end of the day. And I think that the general population, or the population that's interested in sports, will gain from understanding what that really means, and also in their attitude towards DSD and their attitude towards sports and fairness. Because, yeah, it's just, at the end of the day, those are people with conditions that they didn't choose to have, and it's not necessarily a bad thing. So yeah, and I think, and my other main thing that I want to make sure got across is that DSD and transgender, those are two entirely different situations, and I think that commonly they're grouped together, and that's not fair to either of them, right? With differing reasons for their circumstances. And yeah, I just think that the more knowledge that anyone has, the more they can make an informed decision that is the best for them and suits them the best. So people that are, involved in athletics or involved in sports are very interested in, a lot of coaches and stuff, that they just want to understand, like, the recent regulations. What do they mean? Is it fair? Is it not fair? How much of it is evidence driven? How much of it is political? So I think that I've come across a number of people that are just really interested in understanding what the regulations even mean. Where did it come from? So yeah.
Right. And if it affects the athletes, it's good to understand how. And because when you see cases in media, you make up your mind on the small part that you understand, and then you say, oh, they should do this, and it's black and white solution. But what you're showing here is that's actually pretty complicated, and to do it right, you need to spend time and do some research and so on. So it's, even with the organizations that are trying to sort it out and are basing their regulations on the science we have at the moment, even these organizations are not perfect because it is so complicated and hard to do.
Exactly. Yeah, well said.
All right, two short questions to finish. If you could make one change in sports on the base of your research, previous or the research you do now, what would it be?
I don't think that I have any specific change today or tomorrow that I would implement based off of what we know, but I do think that understanding that putting two thresholds, an SRY threshold and a testosterone threshold on performance is just a simplistic view, and I think it'd be good to appreciate the complexities of sex development and how there's more to it than just those two things. So I think mainly it'd be to have the data supporting it. And so to have, I think I mentioned before, to have, again, there's nothing I would change today or tomorrow because we just don't have the data yet. But to be able to have the data and the evidence to choose a threshold for testosterone, to choose maybe a receptive level or sensitivity to the testosterone, and maybe to just have the information to support that threshold, that whatever one ends up getting chosen. And, if there are individuals that fall in between because of the SRY test, to have an avenue for them and for it not to be black and white for those individuals that don't fit into that black and white category, so that they at least have some clarity. But I think at the end of the day, I feel like I'm very repetitive, but at the end of the day, like, I don't have any specific changes I'd make. I would just like to see things be supported by robust science, and for that to be the driver and not for a political agenda or anything else. And not that I'm saying that that is what's happening, but just that it's hard for a scientist to see when, decisions beforehand were made around the different thresholds, and people studying endocrinology and knowing what we know, we're like, where are these thresholds coming from? Like the evidence to support it, especially the testosterone threshold, are pretty weak. So more evidence-based decisions, or guidelines, let's put it that way.
All right, and the last question to finish is where people can find you if they want to look at your research, get in touch, look what you're up to.
I'm not very active, but I do have a LinkedIn account. That's my first and last name. And our research, I mean, anywhere that if you search Google Scholar or PubMed, then any peer-reviewed publication comes out there.
So thank you so much, Shani. Huge pleasure to talk to you. And thank you for agreeing to talk about the topic that is hot and very debated at the moment. Thank you.
Thanks, Alex. It's been great again speaking with you.